Okay, welcome everybody to the last Validate event of 2025, which is very exciting. I assure you we have plenty of plans for 2026, including a continuation of these seminars. So just a background, this was, we had a workshop, a very successful workshop in Bogota back in May. And the talks, including the ones you hear today, was of such high quality, we thought that we, if possible, could share them with the Validate network and community. So we've asked them to sort of update and tweak, but just give their talks to everyone in Validate. And so this is the last of this year, but as I said, we'll hope to have 2 more ready next year. I'm about to put them in the calendar. You'll find out by the end of the week that we'll have 2 more skin NTD seminars next year, early next year. So with that, I'm just going to introduce our first speaker. Now, Rajko is going to speak, and then we're going to do a Q&A. So I'm going to introduce Rajko first. Professor Rajko Reljic is the Professor of Immunology at City St. George's University in the UK. His TB work involves the use of several mucosal vaccine delivery systems, including nanoparticle, inactivated bacterial spores, liposomes, and self-adjuvating recombinant immune complexes. As you can see, I'm not a science graduate, but and I'm going to hand over to Rajko for his talk. Thank you very much, Rajko.
Thanks very much, Blakely. I'll first upload my presentation. Is that visible?
Yes.
You need to switch them around again, Rajko. Yeah, was it display settings and then there we go.
Better?
Perfect. Good to go.
Yeah, hi everyone, and welcome to this seminar. Pleasure to talk to you today. I'm Rajko Reljic, as Blakely said. I'm mostly into TB, but I did a bit of a venture into another mycobacterial disease called Buruli ulcer. I'm sure lots of you in the audience are familiar with this disease. And today I want to present you some of our recent work on our attempts to develop a vaccine against Buruli ulcer. So what is Buruli ulcer? It is one of those neglected tropical diseases. It's caused by a bacterium called Mycobacterium ulcerans. So it is from the family of Mycobacteria, but a completely different manifestation of disease compared to TB. Now, why do we call these type of infections neglected tropical disease? This is a term which is used very lightly, but in fact there are some stricter definitions. The term neglect is used because these diseases affect people who are underserved and marginalized, lacking access to basic services. This results in neglect of these diseases in terms of political prioritization and resource allocation, and are almost ignored by global funding agencies and industry. I'll come back to this particular statement at the end of my talk. As a result, vaccines for them do not exist, and for some, not even specific treatments. Now, I do apologize for a rather gory picture, but trust me, if I was a fully fledged Buruli ulcer physician, I would show you some much more unsavory pictures, which unfortunately this disease can result in, but you get the idea. So this is a disease of the skin tissue. Essentially, the skin tissue is completely decomposed, eaten up by the infection, forming ulcers which can expand and become very, very large. So it's one of those diseases where mortality is not very, very high, but morbidity is incredibly high. As you can imagine, it's a very unsightly appearance. So these lesions can appear pretty much everywhere around the body, but as you can see on this diagram here, some locations are more frequent than others, particularly the limbs, lower limbs, also arms, less so the main body. Epidemiology of BU is shown on this diagram here. So it's been found in about 33 countries around the world, with about 5,000 cases each year reported in 15 out of these 33 countries. Most patients are children aged under 15. And as I've already mentioned, although disease carries a relatively low mortality risk, nothing compared to TB, for example, it bears a huge morbidity and economic burden, with costs of treatment rising as high as $30,000 in some cases. So main hotspots are Western and parts of Central Africa. And the other hotspot is Australia, and then also some other countries, but with relatively lower prevalence. The main hotspots are West Africa and Australia. What transmits this mycobacterium? Obviously, the human is the host, but there are some other hosts, including some of the domestic animals. In contrast, there are many potential and proven vectors for transmission of Mycobacterium ulcerans, and as you can see them here indicated, so a plethora of different organisms, insects, and so on, most of them water dwellers that are suspected of transmitting the infection. I must say there is no clear-cut evidence about the mode of transmission, so these are just either circumstantial evidences or best guesses. The disease can be treated, but the treatment until up to recently had been rather complicated. It included lots of injectables, which is never ideal. But my colleague Mark Wonsborough-Jones here at St. George's and collaborated in Ghana, Richard Phillips, with colleagues, developed a new treatment based on oral drugs, rifampicin and clarithromycin, which has been proven to be very efficient, and this is now officially WHO endorsed or recommended treatment for Buruli ulcer. It still takes several weeks, so it's not exactly a picnic. A little bit about the Mycobacterium ulcerans. So this is obviously one of the mycobacteria pathogens. It produces the most distinctive feature of this bacterium is that it produces a toxin, which is called mycolactone. And this is the only species of mycobacteria that produces mycolactone. And as you know, there are many different types of mycobacteria, pathogenic and non-pathogenic. And it is this toxin mycolactone that causes much of the pathology and disease symptoms in affected individuals. This is the structure of mycolactone, and I'll talk a little bit more about it. But I want to say at this point that WHO recommends treatment for Buruli ulcer, as I mentioned, for several weeks, wound care and prevention of disability, which may include surgery, but no mention of vaccines. And this is something which obviously, in my opinion, needs to change, because the most effective way of preventing a disease is vaccination. And this is what this workshop in Colombia was about, really trying to influence opinions. This particular toxin has many different effects. It's cytotoxic to cells and tissues in the epidermis and in the skin, but it also affects immune cells. And this diagram here proposes many different mechanisms of immune suppression by mycolactone, as you can see, including all types of cells, including lymphocytes as well as myeloid cells and neutrophils and so on. So this is a very potent toxin that exerts many different activities. So I already mentioned we need to try and develop a vaccine, and that is not to say, that's not to say that there haven't been efforts to do so in the past. There were many historic and more recent efforts to develop a vaccine, but unfortunately nothing succeeded, and nothing ever got to licensure. So I just grouped previous attempt at vaccine development for Buruli ulcer into two categories. So whole bacteria, it was a preferred approach historically. And here people used either BCG, which of course is a related Mycobacterium and the current vaccine for TB. They used Marinum, which is an equivalent to Mycobacterium ulcerans that lives in the sea, but or in water, but it doesn't produce the toxin, which makes it safer to use in humans. And then even Mycobacterium ulcerans itself at low doses, or even strains of Mycobacterium ulcerans with the mycolactone toxin in some way disabled. But as you can see in the summary, none of these resulted in particularly great success. Then the next attempt was develop subunit vaccines. So this would be protein-based vaccines. Of course, there's a range of antigens to choose from. Mycobacteria has 3,000 antigens, and some of them are listed here. Again, combined with different adjuvants, or even used as a prime boost regimen with whole organisms. But again, nothing resulted in a very promising vaccine candidate for Buruli ulcer. So we're pretty much at the square one. There is no licensed vaccines, and that's not for the lack of attempts. But my provocative point here is maybe in all these historic efforts of trying to develop a Buruli ulcer vaccine, we have been looking in the wrong places. This molecule, mycolactone, is clearly critical to the pathogen and establishment of infection and disease, and it obviously modulates immune response. So why exclude it rather than in some way include it in the vaccine formulation in a way that would be acceptable as a vaccine formulation? Obviously, we wouldn't want to inject into anyone large amounts of this toxin and cause local disease, but maybe with careful selection of the concentration and inclusion of various other components into the formulation, this could be acceptable for human vaccination. So these are the questions that we asked. Is mycolactone potentially immunogenic as well as immunosuppressive? We know it's immunosuppressive, but is it immunogenic? The problem with this is it's not a protein, it's a polyketide. So there are no classical T cell responses, but potentially unconventional T cell responses could be developed. Also, no antibodies have been detected against this organism, but there are monoclonals that have been made in vitro when the mycolactone was displayed on another protein. But importantly, mycolactone could calibrate immune responses to protein antigens so that they better reflect those induced by natural infection. In other words, if we induce immune responses to selected protein antigens in presence of mycolactone, just as is the case during infection, maybe those responses will be more relevant or more protective. And then we also asked whether enzymes involved in the synthesis of this toxin, and there are many of these enzymes, 9 of them, could be protective themselves. So therefore, there is a case for including mycolactone into vaccine formulations. The case against mycolactone is a toxin responsible for much of the BU pathology, and so obviously it could potentially cause local disease. However, as I already mentioned, a controlled low dose in a vaccine is likely to avoid toxicity while better calibrating the induced immune response. So we teamed up with people in Kumasi in Ghana, and most of the work, I should say, what I'm presenting today is done by Dr. Justice Kofi Appiah, who was a clinical PhD student in my group. Also, Professor Richard Phillips, who is the head of the Kumasi Center for Collaborative Research in Tropical Medicine. And we set out several aims. First of all, to establish an experimental model of Mycobacterium ulcerans infection, and these were mouse footpads. And then to test if additional mycolactone, which has never been done before, may induce more protective immunity. than protein antigens alone. And then also compare protective efficacy of live attenuated strains and our subunit vaccine formulation, and also to test prime booster regimens combining both. So to test our vaccines in these experimental models, we had to develop an experimental model which physiologically reflects progression of Buruli ulcer disease and therefore is relevant. So in humans, progression is indicated here. It starts as a small papule, which then becomes a plaque, eventually oedematous form, and the final stage is ulceration. So could we reproduce that in mice? Yes, we could to a large extent. So this shows infection of mouse footpads with this organism. This is at the beginning, obviously no change to the footpad, but as you can see through these various grades, 1 to 4, the footpad begins to swell, swell more, and eventually at some stage it begins to ulcerate, and this is then affecting the whole limb rather than the footpad. So this is the model that we developed. Of course, we didn't need to go to this final stage. We ended up stopping at this stage here, which I'm pointing, so grade 2, because this was sufficient for us to assess vaccine performance and also ensure humane endpoints and no sufferings to animal. So first of all, we developed the model. As you can see here, one footpad acted as a control, so non-injected with bacteria. The other was injected, and as you can see, footpad got swollen. If you inject different numbers of mycobacteria, of course, you get more and more swelling. In some instances, too much, so the animals had to be humanely put down. But then one interesting observation that we've noticed is that if you inject a relatively large dose of mycobacteria, the swelling goes up and then it pretty much stays up and unchanged over the weeks. But if you inject a small amount of bacteria, the swelling starts, but then over the weeks it begins to self-dissolve. So in other words, this suggests to us that when mycolactone is produced at low level, then immunity may indeed develop and resolve the swelling. And this was a further, a support argument for inclusion of mycolactone into our vaccines. We needed to, of course, determine the safe concentration of mycolactone that can be included in formulation. So we did cytotoxicity in vitro against a number of different cell lines, including epithelial cells, myeloid cells, primary splenocytes, and so on, fibroblasts. And generally at around 10 nanograms per milliliter, or maybe 5 to 10 nanograms per milliliter, it begins to be cytotoxic to the cell. So ideally, we would have liked to stay in the range below 10 nanograms per milliliter. So I'll park mycolactone for the moment and just focus a little bit on the enzymes that are responsible for synthesis of mycolactone. So these enzymes are present on a particular plasmid in Mycobacterium ulcerans, very large plasmid, which has several modules responsible for synthesis of essentially 10 different enzymes that all contribute towards synthesis of mycolactone. As you can see here in different colors, these different enzymes are responsible for different steps in the synthesis. We also, we didn't express all of these enzymes, but we expressed 5 or 6 of those. You'll see them. We also, of course, included some of the more mainstream Mycobacterium ulcerans antigens, such as antigen 85, which is responsible for cell wall synthesis, because these are obviously known as very immunodominant and potentially protective antigens. So here I'm showing expression of these different antigens. I'm not go through each individual one, but we express them in E. coli and purify them to high degree of purity. And then we did a preliminary experiment to test their immunogenicity in mice, combined with a standard adjuvant, in which case we used QuILA. So there is the antigen 85, for example, which gave a very nice antibody response. There are some of these enzymes, in particular KRA stands for ketoreductase A also gave very high antibody responses. ATP enzyme also good responses, weaker responses for some of the other enzymes. When we looked for cellular immune responses, a similar pattern was observed. So when we looked specifically, for example, for production of interferon gamma, which is important for protective responses against this disease, you can see that antigen 85 and KRA induced the best interferon gamma production, much superior to other protein antigens. This was also true for IL-17, which again is important for intracellular pathogens. And so this led us to then down select antigen 85A and KRA as the two preferred antigens because they were the most immunogenic. And this is essentially a summary of our rationale why we included these two antigens into our vaccine formulation. We also looked at the proportion of CD4 and CD8 T cells that they induce, and as you can see here, they formed quite well. So another component or another player in this protocol was a strain of Mycobacterium ulcerans that cannot express functional mycolactone. It lost mycolactone through various manipulations. This strain is called M51, or we just call it MU delta. And we can actually clearly demonstrate that this strain cannot make mycolactone because in red, I'm showing here that wild-type bacteria produce supernatant that was very cytotoxic to cells. Media alone, of course, is not, and the mutant strain is not either. So those are all the players that we considered to test in various combinations. And so we designed an efficacy study which included these 5 different formulations. I'll go through them, and then some additional formulations in the second arm, which were subcomponents of the vaccine formulation to see whether there's any redundancy and so on. Of course, we couldn't test every single possible combination. We would require probably 15, 20 experimental groups, but we tested at least some. So our baseline was just mock immunized animals. We used BCG as a reference point because we know it's partially protective at least. We used this mutant of Mycobacterium ulcerans, which has also previously been shown to be at least partially protective. And then we used our vaccine formulation, which we termed Buruli, and we use it either in combination with the mutant strain or on its own as a homologous prime boost regimen. So what is Buruli? It's a formulation that contains mycolactone. It contains these two selected antigens, and it contains Quil-A as an adjuvant. As for the 2 live bacteria, as vaccines, we use BCG and mutant strain 10 to the 5 colony-forming units per vaccine dose. So we immunized these animals according to these regimens, and here we analyzed, first of all, cytokine profiles. And as you can see here, for example, control animals showed very low level or no any of the cytokines except for IL-6, which went a little bit funny. But as you can see, vaccine formulations then induce different levels. Interestingly, the last 2, which is either Buruli on its own or mutant plus Buruli as a boost, always induce the highest levels of these cytokines, including IL-10, which we seem to now believe is quite important to correlate of protection in Buruli disease. So those were responses to antigen 85 antigen. These are responses to KRA antigen. And again, very, very similar outcome. The strongest responses were seen in the formulation that received Buruli formulation. The most important test of these vaccine formulations was the actual ability to suppress swelling of the footpads. and essentially prevent disease in these experimental animals. And here we are showing outcomes for each individual animals within each experimental group. And what we are showing here is % change in the footpad size. So you can actually measure footpad size by any diameter you want to and record it. And as you can see, in control animals, the size of these things was more than 60% increased footpad. Different vaccine formulations reduced it to different levels, but the two Buruli formulations, either alone or in combination with the mutant strain, completely prevented disease of swelling in these animals. And in this sub-arm or arm of the experiment, we tested different individual antigens, for example, just one antigen or the other, or mycolactone alone. We did it in combination with mutant strain as the prime and then boosting with these formulations. And again, same outcome, of course, Buruli completely prevented swelling, whereas others suppressed it to a large level, but not completely suppressed. Do I have 5 more minutes, Blackley? Or 3 more minutes?
No, you got a few more minutes. We have some
Okay,
padding,
cool.
so yeah.
This is the most important readout, though. So this shows here % of animals that were completely protected against infection in Mycobacterium ulcerans. So this is no longer a case whether the animals had more or less swelling, because that means that the infection wasn't prevented, but rather that there was no swelling at all. So this plots the number of animals with no swelling at all. And as you can see, no animals in the control group, very few animals BCG. The mutant strain did pretty well, but the best outcome was the two Buruli formulations, where essentially 100% animals were protected completely from infection. There was no swelling in any of the animals used. We also looked for the bacterial presence in tissues. This proved to be a bit tricky. We did a bacterial DNA from the footpad. Culturing Mycobacterium ulcerans is very, very difficult. So we did DNA. And as you can see here, only control animals showed significant amount of DNA in the tissue. All of the formulations showed very little or no DNA. Likewise, when we measured CRP, so this is C-reactive protein. This is a measure of inflammatory response. Again, control animals had high inflammatory response, vaccinated animals much lower. So that's the end of our experimental evidence, and I'm just gonna go very quickly through our conclusions. So mycolactone is cytotoxic and immunosuppressive. However, it can be immunogenic if used at the right dose and with an appropriate adjuvant. It can also serve as an immune modulator to calibrate immune response to protein antigens. In our hands, the mutant, so not able to express mycolactone, showed to be quite a decent vaccine candidate, in fact, more so than in some other studies. But consistently in all our experiments, our formulation, Buruli formulation, was always the most protective vaccine candidate, providing 100% protection. And so we therefore believe that Buruli is a promising vaccine candidate for this disease. So what next? Well, there are a number of potential obstacles that we need to address. So vaccines are not a priority area for BU, unfortunately. WHO supports work towards 3 pillars, but no mention of vaccines. So understanding the mode of transmission, developing rapid diagnostic tests, and establish best case antibiotic treatments. Other obstacle potentially could be that our composite vaccine formulation might not lend itself very well to large-scale production. Obviously, it's a little bit complex. There are potential issues with toxin in formulation, safety concerns, real or perceived. However, at least in our experimental studies, a controlled low dose is very unlikely to cause local pathology, and we observed none in mice at the site of vaccine injection. And really the biggest obstacle is lack of interest from industry, because they don't see that they would have return on their investment, because they would not be able to sell enough doses. Of course, this is a relatively a small endemic disease. So then why do we bother developing this vaccine candidate? being a preventable infectious disease, this should not stop the scientific drive to develop a vaccine, with the view that it may be easier to change policies, views, political will, and commercial priorities once there is a promising vaccine candidate rather than when there is none. And with that, I'm done. I'm just going to acknowledge a number of people that participated in this study, in particular Dr. Justice Appiah, who was, as I said, PhD student, also some other people in my research group, and also external collaborator, in particular Richard Phillips. Chris Hagen provided us with lots of advice, and also Professor Tim Steiner, who provided us with genetic constructs for the mycolactone enzymes. So with that, I'll finish and would be very happy to answer any questions. Thank you.
All right. Thank you for that, and thank you so much for keeping so closely to time there, Rajko. It's a rare gift. So do we have any questions? As I said, we already have a hand raised. Sato, I think I know your name, but if you can tell me who your name is, please go ahead.
Blakely, this is Dr. Abhay Saraskar from Ohio State
Yes.
University from US. So very nice talk. So I had a couple of questions. So first one is, my understanding is that this infection resolution does not induce long-term immunity, right? These people can get reinfected again, unlike many other intracellular pathogens. So what do you expect if you have a vaccine, you know, what would be the strategy for vaccinating people, you know? And the second question is that if mycolactone is the toxin which causes the pathology, why can't we think about just doing a vaccine which is like tetanus toxide, where you take the whole toxin, you know, inactivate and just try to reduce the pathology rather than infection? Because if someone is infected, you would nevertheless need treatment, whether you develop pathology. I think pathology is the main part, right? If you can prevent the ulcer by, you know, like tetanus toxide, where, you know, you get Clostridium, but to prevent mortality by neutralizing toxin.
Yeah, these are all very good points. So to address them, I'll go first to potential for reinfection. To be entirely honest, I don't know what's the incidence of reinfection of Buruli ulcer. As I said, I'm not a fully fledged Buruli expert, but I would assume it is not very, very, very common. It probably, if it does take place, it's probably something that may occur in some individuals because of dysregulated immune system or whatever. It would be a pretty bad outcome if that were the case for a vaccine, because obviously, you know, a vaccine does need to induce a long-lasting immune response. As we know, unfortunately, many infections can also reoccur, even though there's an immune response. Just consider, for example, chlamydia infections and things like that. So, but I would say, you know, I'm not exactly an expert on epidemiology and the rate of reinfection. Mycolactone itself is a toxin. If you're thinking about potential treatment that would target mycolactone, I'm not sure how that would work, but in terms of using attenuated mycolactone as a component in vaccine formulation, that's certainly on. I don't know myself how I'd make attenuated. I'm not an organic chemist, but there is a clearly potential to develop attenuated form of or inactive form of mycolactone, which then may be a safer option for human vaccination.
Thank you.
I hope that answered your question.
Yeah, great. Thank you.
Thank you.
Thank you, Abhay. Do we have any other questions, either in the chat, or you can raise your hand using the react button? It usually means you've answered all their questions in the talk. Yeah, so I'm going to give it a few more seconds. Any questions? Okay. Oh, one, Elena, go ahead.
Hi, Rajko. That's a great talk and fantastic data. I was wondering if you have any evidence of how your vaccine would work without mycolactones. For example, if you were to use a mutant plus antigen 85A plus KRA and QuIL-A without the mycolactone, how would that compare?
Well, as I said, we had the hi Elena. So we had a subcomponent of this experiment where we tested individual components, and as I showed in this particular graph here, if you sorry, not that one, one more. So if you look at this part here, if you just use, for example, one or the other antigen without mycolactone, you get pretty good protection, but not as good as the complete formulation. Now, the problem with this is we used mutant here to prime animals and then boost with these individual components, and I think we oversold ourselves in order, to see what components contributes how much to the protection. So in short, I take your point. There is still plenty to be done in terms of determining what is the minimal essential formulation necessary and sufficient for protection, rather than cramming as many things as we possibly can. So it's all about detecting potential redundancies, and we still got some work to do.
Thank you, Elena. And to give people 10 seconds to raise their hand if they want to ask a question. I think that might be a good time to move on. Any questions? Okay, so Rajko, thank you so much. I think Rajko has to leave now, so if you would like to express your appreciation by just waving or sending a message in the chat, that would be great. But thank you so much for your talk. Yep, applaud from Elena. Applaud from Lydia. There we go. Thank you so much. And now I'm going to introduce our next speaker. If you could stop sharing, Rajko, that'd be really helpful.
Yes.
Yeah, lots of applauses. I can't name them all. Thank you so much. So everybody, we're going to introduce our next amazing speaker, who is, if you think my pronunciation of science words are a butchery, that will listen to my attempt to pronounce some Spanish ones now. Camila, sorry, Camila, is it Camila Indias de Oliveira? Is that good enough? Dr. Camila de Oliveira is a researcher in Cruz in Brazil, where she leads a group interested in host pathogens interactions, focusing on leishmania. Her research area includes the pathogenesis of cutaneous leishmaniasis and the development of novel treatment alternatives and immunoprophylaxis. And I'm going to hand over to her. If we're still on, is this your slides? Is this your view? Okay.
No.
I'm going to make sure that, bear with me. Let me just, I think I can ask, if you could stop sharing, Rajko. Oh, no.
I can send a request for you, I think.
Sorry, I'm back. Stop
That's
sharing. Where is the option of stop sharing?
I should have to do it myself, but the option not seem to be coming up. I might be editing this about the final video. Hang on.
I think I can share.
There we go. You should be able to share now.
Okay.
There we go. Yeah, and we can see the back end. And if you start there, and
now
you need to switch around
swap,
again.
Right?
You need to swap again, yeah.
Swap. Yeah, now you have it, right?
So over to you, Camila. Thank you very much.
Okay. So thank you very much for having me. It's good to be here, speaking from Salvador in Bahia. It's quite sunny and lunchtime for us. So I'm going to be talking about attenuated parasites as a road for vaccine development against cutaneous leishmaniasis, and I'm going to be focusing on cutaneous leishmaniasis caused by Leishmania brasiliensis for the first part of the topic, and then for leishmaniasis in general. So I'm going to use Rajko's talk about neglected diseases, because leishmaniasis falls in the same category. It's caused by Leishmania, which is a protozoan parasite with an intracellular lifestyle. There are more than 20 species that cause disease in humans, and it's transmitted by different sandfly vectors. There's a spectrum of clinical manifestations depending on the parasite species and the host response. And here in this cartoon, I'm showing the typical ulcer that you get from infection with Leishmania major that occurs in the Middle East, mostly in parts of Africa, and with Leishmania brasiliensis, that is the main species causing cutaneous leishmaniasis here in the Americas, or in Brazil specifically. So there are many countries that are endemic for leishmaniasis. You see here in this map, all the countries that are labeled in red. So it occurs all over the world. And there's a increased risk of transmission and leishmaniasis occurrence because of novel risk factors, so to speak. So one staying risk factor is socioeconomic conditions and malnutrition, but there are novel risk factors associated with population mobility and environmental and climate changes, for example. So the available treatments for cutaneous leishmaniasis, and I'm speaking here because of the prevalence of Leishmania brasiliensis in Brazil, there are some treatments that are available. Some of them are very old, like the pentavalent antimonials. But what is important for me to highlight here is that the options for treating cutaneous leishmaniasis have changed very little over the years. There's a possibility of using intralesional treatment, but it's mostly intravenous antimony that is available. The drugs remain expensive and they present important side effects. And in Bahia here, where we work in the endemic area, there's around 50% treatment failure that has been recorded. So there's a very urgent need to either develop new drugs or to perhaps think about developing vaccines. So the feasibility of developing vaccines resides in three mostly broad areas. There's the practice of leishmanization, which is a very old practice that is associated with a deliberate infection with mainly Leishmania major that has been shown to provide long-lasting protection against cutaneous leishmaniasis and can be protective also against visceral leishmaniasis. So this has been used in the past in the Middle East and the former Soviet Union, but it has been stopped due to safety concerns because there are cases of non-healing lesions and there isn't really a standardized Leishmania major strain that can be produced in large scale to be used in this kind of practice. There's evidence of natural immunity that does develop following a primary infection. So people that recover from a cutaneous leishmaniasis, they have protection against cutaneous leishmaniasis and also against visceral leishmaniasis. There are studies showing that. People that develop visceral leishmaniasis, on the other hand, which is another important form of the disease, they develop lifelong immunity in most cases. And also many infections, they can remain asymptomatic. So that also demonstrates durable immunity to the Leishmania parasite. Based on these findings, there are many papers in the literature showing the development of experimental vaccines in different models, a lot of them in mice, but also in dogs and in primates, showing that you can have a strong cell-mediated immune response and protection against infection. But I want to highlight that in these experimental vaccines and in all this literature, there's an important problem regarding the model that is used to address protection, different species and different models, as you can see here, but also the correlates that are used to define then whether a candidate is protective or not. But putting all of this together, what we can conclude is that there are multiple lines of evidence that support the feasibility of a human vaccine against leishmaniasis overall. So back to the strategies used to develop a vaccine for Leishmania, there are many of them in place that have been used in these experimental demonstrations. There's DNA vaccines, antigen cocktail vaccines, fractionated Leishmania parasites vaccine, recombinant protein vaccines, or killed parasite vaccines as well. But I want to focus today on the use of live attenuated vaccines. So that would be parasites that you can attenuate in the lab, either by chemical attenuation or by genetic modification of the parasites. And I'm not gonna go over this literature of how you can attenuate the parasites, but I want to show you the findings that we have once we use these attenuated cell lines for immunization. So what we know is that when you have vaccination with subunit vaccines, and this is for different pathogens, You can use different types of preparations and different strategies using prime boost strategy or the use of adjuvants. What you do get is the development of effector T cells and memory T cells that enclose limited immune response. So these effector memory T cells are short-lived and they disappear, but you do get the memory T cells that stay for a long time. And once you get a virulent challenge, then you get sort of a delayed response. They take more time to induce the protective immune response. So in terms of vaccination with subunit vaccines, you have very little safety concerns because they're purified proteins or peptides or recombinant proteins, but you also have lower efficacy. But once you work with live attenuated vaccines, on the other hand, and this has been shown with Leishmania parasites, with sporozoites from Plasmodium, or with Trypanosoma, and this can be attenuated by genetic modification or chemical exposure, then you have a pool of effector T cells and memory T cells that showcase population-wide immune responses. And once you have antigen re-stimulation, then these populations, the effector and the memory T cells, they expand rapidly. So you have a rapid response from the effector T cells, but you can also have a second phase of response from the effector T cells that derive from the memory T cells. So in terms of the efficacy, you have more efficacy with these attenuated vaccines, but you also have to control for safety concerns because these are attenuated parasites and there's issues regarding reversal of virulence and also issues regarding use of these attenuated vaccines in immunosuppressed individuals, for example. So using this strategy of attenuated vaccines, what we know is that there's a very large body of literature addressing the use of attenuated Leishmania parasites for the development of vaccines. And I just want to highlight that a lot of the literature has been produced in Leishmania donovani, that is a species associated with visceral leishmaniasis, or Leishmania major, associated with cutaneous leishmaniasis, and there's very, very little work done with Leishmania brasiliensis, that is the main species causing cutaneous leishmaniasis here in Brazil. I also want to highlight that a lot of this work, as I mentioned before, has been done in mice, mostly in bulb C mice because they're a good, or B6 mice because they reproduce the paradigm that you see regarding Th1 and Th2 responses once you infect with Leishmania major. But there's very little work with monkeys that would be the most relatable model if you consider diseases in human. So there are a lot of antigens that have been approached for the gene targeting to induce attenuation or to develop attenuated cell lines. And among these, I have put here on the right the promising gene targets or the promising candidates that have been shown in different publications. And I want to highlight here centrin as one of the models that we're going to be showing or exploring more in the rest of this presentation. So centrin is a protein associated or important for the multiplication of the Leishmania parasite. And once you delete this gene from the Leishmania parasite, what happens is that the parasite cannot replicate fully or cannot replicate productively. So what happens is that in Leishmania parasites lacking the centrin-1 gene, what happens is that they develop multi-nuclei, but they don't fractionate. So you have a large cell with multiple nuclei, and the cell eventually dies, and you don't get the full infection or the reproductive infection within the macrophage. On the other hand, the promastigotes, that are the life forms that you can propagate in culture in the lab, they work very well, they can develop very well. So you can culture these parasites in the lab, but once they infect the macrophage host cell, they cannot divide. So in this case, you have a parasite that is very good to grow in the lab, but once you infect with this parasite, they cannot sustain the infection, so the host doesn't get a clinical manifestation or doesn't get disease. So there's a lot of studies that evaluated centrin-deficient Leishmania parasites as vaccine candidates. These have been evaluated in Leishmania major, in Leishmania donovani, in Leishmania mexicana. And I just want to highlight here the results that are usually shown in this paper that showcase then the protective efficacy. What you get is that an immunized mouse that is challenged with Leishmania, they don't develop disease. You have no lesions, as you see here on the graph, and they're completely protected from infection, as you can see here on the mouse ear. The parasite load is also significantly reduced both at the infection site and in draining lymph nodes. And this protection is associated with the development of CD4 T cells that are producing interferon gamma, which is the hallmark cytokine associated with Leishmania, with protection against Leishmania. So all of these works have shown to different extents that Leishmania parasites lacking the centrin gene, they are protective because they can induce the development of this type of immune response, Th1 biased, and they don't cause disease once the host is immunized, and this has been shown in different models. In terms of natural infection, what has been shown more recently is that immunization with Leishmania major lacking in centrin confirms long-term protection in dogs in natural settings, showing then that you can apply this type of vaccination or this type of strategy in hosts that are living in endemic areas, which would be an important evidence that you can approach this for larger trials, including humans. So based on all of this literature, we started a long time ago to evaluate whether centrin-deficient parasites would be also efficient at conferring protection against Leishmania brasiliensis. And I'm going to summarize the results that we have in the coming slides. What we did was to develop a Leishmania brasiliensis lacking centrin, and we show that this cell line that behaves just like the other Leishmania species lacking centrin. They don't form productive amastigotes. And once the macrophage is infected, it develops then these many nuclei, but cannot sustain the infection. And we use this cell line to address whether immunization with this attenuated cell line would induce protection in a model of Leishmania brasiliensis infection. And I just want to highlight here that the model that we have, it includes infection in the dermis of the mouse ear with Leishmania brasiliensis. We use biopsy mice, and they develop this lesion that is ulcerated. It's very much close to what we see in the human host, and it's a self-healing lesion. So we have this kind of time frame to evaluate whether immunization with this attenuated cell line would confer protection or not. So to our very big disappointment, what we observed is that, and I'm not gonna go over in detail because these results have been published, but to our disappointment, we observed that once a mouse is immunized with the centrin-deficient Leishmania brasiliensis, is later challenged with virulent parasites here, what we see is that the challenged mice, they develop productive lesions, shown here in orange, very much similar to what you see in a naive mouse that has not been immunized. In comparison, a mouse that has been leishmanized or that has been infected with Leishmania and has healed a primary infection, they are completely protected, which would be the gold standard for a protection in this model at least. So this was very surprising because this is the first time that we showed that a Leishmania species lacking centrin would not be able to confer protection against a homologous challenge. And you can see here on the right also that the immunization with the centrin-deficient Leishmania brasiliensis induces productive lesions, not as bad as the naive mouse, but still you have a detectable lesions, whereas the gold standard, the leishmanized mouse, a mouse that has healed a primary infection, they're completely protected. But this was not the end of it, because we were able to show then that if you use another Leishmania species lacking centrin, which in this case was Leishmania donovani, you get complete protection. So immunization with this centrin deficient Leishmania donovani followed by challenge with Leishmania brasiliensis induces complete protection, as you can see here in the blue line, whereas the other case, Leishmania brasiliensis, does not confer protection. And this protection with Leishmania donovani, lacking centrin, correlates with a significantly lower parasite load at the ear and at the draining lymph nodes. So we couldn't get protection with Leishmania brasiliensis, a homologous challenge, but we could get it with a heterologous challenge with Leishmania donovani. And this was interesting for the scientific purposes, but it was also showing then that we can work towards a vaccine that can possibly be used against many different species, which would be much more feasible than having to develop a vaccine for each species, for each country, or something like that. So this is just a summary showing that immunization with Leishmania major, that I have not shown here, but with Leishmania donovani, lacking centrin, it induces an effector T cell response that is characterized by CD4 T cells producing interferon gamma and very low levels of IL-10 and IL-4, whereas immunization with Leishmania brasiliensis lacking centrin induces a more mixed response, but we don't get a predominance of interferon gamma, which would kind of explain why we don't have protection using this specific attenuated cell line in Leishmania brasiliensis. So centrin deficient Leishmania brasiliensis cannot confer homologous protection, but cross protection is possible, and Leishmania major has advanced as a vaccine candidate in regulatory steps, and I just want to show this here. So we have 3 vaccine candidates that are advancing towards clinical trials. We have, and one of them is Leishmania major lacking in centrin. So based on this advancement in the clinical trials, what would be the hurdles for actually advancing towards using this in the field? So one of the important hurdles that we have once we have a vaccine candidate that can be used in the field effectively, one of the hurdles is evaluation of immunity in vaccinated individuals. So once an individual is immunized with this candidate, how do we know it has developed a protective response or a response to the vaccine itself? And another problem later on would be to determine the efficacy of a Leishmania vaccine, because in general, disease development is the traditional endpoint for vaccine trials. And this was very well demonstrated in the COVID vaccine trials. But for Leishmania, this would be very difficult because there are because of elimination programs in place, especially in India, which would be the most advanced elimination program. You have very few cases for detected. So this would make these evaluations very long and very costly. So this would be another important hurdle to be overcome. And also we need a surrogate marker of immunity to determine vaccine efficacy. What would be the readout once we have vaccinated individuals, once they're in the endemic area being challenged by live parasites? What would be the surrogate marker that we could use if we could not wait for natural development of the disease because of these endpoint efficacy difficulties? So one of the things that we began working on was the evaluation of a Leishmanian skin test, or LST for short, which we'll be using from now on. So the LST is a type 4 cell-mediated hypersensitivity test. It involves the inoculation of Leishmania antigens to induce a DTH response in individuals that have been exposed to Leishmania, either because of a primary infection or have been vaccinated, for example. And it becomes positive then following this natural infection, which is associated with long-lasting immunity, especially in VL. So the LST conversion or positivity to do the LST test would be an important endpoint for protective immunity in trials involving vaccine evaluations. And this has been used with heat-killed parasites and also for the identification of immunity to Leishmania and identification of individuals that have been exposed to Leishmania. And there are two main cases that we can talk about for LST. One is for is the detection of latent infections for surveillance studies, for example. So you go to the field and you administer the LST test, and you can then see who has been exposed, who has immunity to Leishmania by use of the LST test. Or you can also use it for a vaccine to correlate vaccine immunogenicity in cases of vaccination. So once you prepare the LST, you have a good antigen and you have all the development and GMP certified production. You can use it in forms of injection, which is the classical DTH test that would enclose measuring in duration of the test after 48 hours. And this then entails having the test administered to the subject and then having somebody available to have the readout 48 hours later to see if they develop a positive DTH or not. And this can be used in surveillance or in vaccine trials, as I've mentioned. But another important development, once you have this preparation, would be perhaps the use of a quantiferon-like test in which you can stimulate whole blood with this antigen and then measure interferon gamma production. And this, you can scale this to larger trials, and you can do this in the lab or in certain labs. And this would be much easier than having the classical DTH, which needs a person to read the response 48 hours later. So these are the two kind of approaches that you can have or you can develop once you have a GMP certified antigen, which would be the Leishmania skin test for the development of the LST test. So there is major advances in this area. There's an LST test, there's a Leishmania antigen that has been produced, and I just want to summarize this in this busy slide here. So the antigen preparation has been tested in mice, and you can see that it induces a DTH response here on the left once the mouse has cleared or has been infected, leishmanized, and has cured this primary infection. You have a positive DTH. And then in other studies in hamsters, it was also shown that you can induce DTH responses in mice, in animals that have been exposed to Leishmania, exposed to the Leishmania major lacking in centrin. So this then putting together vaccination with Leishmania lacking centrin with the attenuated cell line, and then testing for the LST test, it is also positive. So now we have an antigen that can be used to ascertain exposure to the parasite in different animal models. And this has been evaluated and developed further in terms of the clinical approved formulations that you could use in humans. And this is work in progress, and the manuscript has been submitted for publication, also showing then that mice that have been vaccinated with Leishmania major lacking centrin, they can develop a DTH response to this antigen, as do mice that have been infected with Leishmania major, wild type Leishmania major. So in the last couple of slides that I have, what we have been doing is assessing the human response to this antigen preparation, which is a GLP grade lyophilized Leishmania donovani antigen preparation. And what we are doing is assessing the response of individuals that are naturally exposed to Leishmania brasiliensis. This has been done here in Bahia, in the endemic area, which is shown here in this map. So Salvador is where Cruz is, where I'm based. And there's a team of physicians and clinicians and investigators that also work in Cruz, with whom we collaborate, that that go to the clinic once a month to see the leishmaniasis patients. And then in this setting, we have observed then whether PBMCs from these individuals collected can be stimulated with this GLP grade antigen preparation, and then if we can detect interferon gamma production in these individuals. So this is just the setup that we used. We tested different amounts of the LST antigen, and we came up with a quantity that would be sufficient to stimulate interferon gamma production. And what we have done is then collect blood from healthy subjects residing in Salvador that is non-endemic for leishmaniasis, from individuals that have cleared a primary infection. They were diagnosed with cutaneous leishmaniasis, they were treated and they healed, and also individuals that have active infection with a lesion. And so blood was drawn from these individuals, and we stimulated PBMCs, and we stimulated whole blood as well with either the GLP grade LST or with soluble Leishmania antigen prepared from Leishmania brasiliensis, so the correlate antigen. And we then evaluated the supernatants for the production of interferon gamma and IL-10 to begin with. So this was just the flow chart showing the individuals. We have close to 20 individuals in each group now for active cutaneous leishmaniasis or healed individuals. And this is the characteristics of these individuals for their clinical characteristics. We don't have significant differences in all the parameters we addressed, except for the majority of individuals are male. And also we have a higher, there's no significant differences, but the males, they're the majority of individuals that we do get in this endemic area. So I want to show the cytokine production, the PBMCs first. What you can see here in the healthy subjects is very low production. But with the active individuals, you get a similar productions using either a Leishmania brasiliensis antigen, shown here in pink, or with the LST GLP grade, shown here in orange. So the LST is recapitulating very well the response that we get in active individuals regarding interferon gamma production, and also in the healed individuals. So we're showing then that we can really use this antigen to probe for interferon gamma production in individuals exposed to Leishmania. And also, not to surprise us, but the LST antigen preparation, it induced also a higher level of IL-10, which we do not see with Leishmania brasiliensis antigen, but this was also expected. In terms of the whole blood stimulation, we also got a very good production of interferon gamma in both active disease and in healed individuals, and again, a significantly higher IL-10 production. So these are kind of preliminary studies that we are doing to prove that this antigen preparation can be used to probe for exposure to Leishmania using either PBMCs or whole blood. And this is important because, as I showed you before, we can think of developing a modified quantiferon kind of assay to be used in the field in larger trials once the vaccine is effectively used to immunize larger cohorts. And then we can think of using this modified quantiferon to probe for immunogenicity and interferon gamma production. So this study is being done here in Bahia, where I'm based, but we've also expanded it now to other endemic areas, to Kenya and Spain. So it's going to be a larger study, including multiple centers, to validate then the use of the LST in multiple settings and in many different kind of infections with Leishmania. So GLP, the LST, GLP preparation can be used as a tool to monitor exposure to Leishmania, previous or active exposure. And we believe we're advancing towards developing an assay to probe for immunity in people vaccinated with the centrin deficient Leishmania vaccine. And it can also be used at some point for surveillance of disease burden in endemic areas. So I want to acknowledge and thank the people involved in the study. These are the students and postdocs involved, mainly Larissa Oliveira. This study is only possible because of the clinicians that we have here in Fiocruz, Edgar Carvalho, Lucas, Paulo, and technical support in the endemic area by Tainan Lago, and also the invaluable partners in this project, Hira Nakasi, Srinivas Ganarvan, Abhay Satoskar, and also the people at Genova who produce the LSD antigen and who will produce also the vaccine. And of course, the sponsors here in terms of financing the study. And I want to end here with a photograph of Salvador that is as beautiful as shown here. So thank you very much.
Thank you, Camila. Sabella and Seema want to say how good your talk was. They said so in the chat if you want to have a look.
Thank you.
Anna Micah, do you have a question at the beginning, at the front of the list? So
yeah, thank you. So first of all, thank you very much for this very nice presentation. In your presentation, you mentioned always that you measure interferon gamma as a tool to measure exposure. And now I've been working on preparation of a whole blood assay for leprosy similarly. And what we found already when we measured interferon gamma, because the idea was also there that we would mimic quantiferon TB. And but what we saw there is that also people that were maybe exposed because they were living in an endemic area, they produced also interferon gamma. So that is why we were not interested in interferon gamma anymore, because it did not give us any discrimination between real infection and those that were only living in an endemic area and may have been exposed. So my question actually is, so first of all, are you considering more other cytokines that maybe can give a better discrimination? And the other thing is, what is your definition of exposure?
So exposure here, what we are defining by is people with a history of leishmaniasis that we know from the endemic area, because we have a cohort of people that we follow. So we have their clinical records and we know that they have been exposed. So these are the healed individuals that we know they have
they have been infected.
Have a scar, and they have a scar, and they have a clinical history of leishmaniasis that is not just mentioned by them, but we have the clinical records. So this is the exposure of the healed individuals.
Yeah, we would refer to that, I think, as infection, then that they have been infected.
Yeah, they have been infected. And the other group would be the people with the active infection at the moment, with the active lesion at the moment of blood collection. So these are the two cohorts that we have. And yes, we want to explore more other markers. So the idea for this first part of the study was just to measure if they have interferon gamma at least.
Yeah.
But we want to make a more comprehensive study, including other markers and larger groups of cytokines and other immune markers that we can test. But we haven't done that yet because of cost. But we want to make a larger panel of markers and perhaps discover markers that can be exclusive to this antigen preparation and not to the Leishmania brasiliensis antigen preparation, for example, if those. But yes, we want to expand, absolutely.
Exactly. Yeah. Well, a lot of biobanking, I hope you are doing.
Yes. Yes.
Okay. Thank you.
Thank you.
Hi. So, Richard.
Very nice talk, and it's great to see that you're producing a GLP antigen. That'll be really useful. I was interested in whether you had explored at all why your centrin knockout didn't behave as an attenuated vaccine as other species have done when they've got the same genetic lesion.
Yes, so I think it's a question of the I think it's a question is too attenuated. We haven't explored more. We've stopped at that one paper, but I think it doesn't survive as long as the other species do. So it dies very early in the mouse. We cannot recover parasites after 1 week of immunization. So I think there's not enough time for the cellular response to develop strongly as it does with the other species. I think that's my main guess.
So it
But we
fits
have
with
to
the
explore more.
idea that an attenuated vaccine needs to remain viable for a while to stimulate the immune response.
Yes,
Yep.
yes. Longer than 1 week at least 2 weeks to 4 weeks, yes.
Thank you.
And Minbera, are you happy to ask a question on camera?
Yeah, thank you very much for the nice presentation. In my experience working on mice with Leishmania atopica, there was interferon gamma production both in control and infected and uninfected monkeys. I think maybe your Leishmania preparation is discriminating that too. Is that?
Sorry, I didn't get the question. Can you say it again?
Yeah. For Leishmania atopica, which is
Yeah.
the species which is common in Ethiopia, and then even in infection, both infected and non-infected individuals was producing interferon gamma to the almost equal level. But in your situation, even for the vaccination, there is difference, is that?
Yeah, so
what was common? My question is, what is common in infection, even just for the infections? There is difference between infected and non-infected in regard to interferon gamma production, like in mice or monkeys?
So for the humans, the individuals, our controls are the individuals residing in non-endemic area. We assume they have never been infected, that they have had no contact with the parasite. For these individuals, there's no cytokine production, is very low. In comparison to the individuals that have been exposed, they have history of leishmaniasis or have an active lesion, for those, the production is much higher. Is that what you asked?
Yeah, I think maybe in our case, it deals with Leishmania atopica. It might have some characteristics.
Differences, yeah. Yes.
Yeah. Thank you very much.
Thank you.
Thank you, Menbarawak. Does anybody else have any questions? Feel free to type in the chat. Sabella says she's looking forward to see what happens in the Kenyan studies. So
yes, we're looking forward to the other studies as well, the other sites. I think it'll be very important as a whole to have these results in many sites, in many countries, and I think we'll have good results.
I think Menbarawak might have another question. Do you have a question? Again, Menbarawak. Yeah. Nope, she lowered her hand. Okay. So I'm just going to give people 10 more seconds to raise their hand really quickly if they have any questions. But other than that, I just want to thank Camila and the now absent Rajko for 2 excellent talks. I'd also like to thank for such a really good turnout today and really engaged audience. So thank you very much. So if you could show your appreciation using the react button or coming on camera and yeah, there we go. There's some applauses all coming up. Thank you very much.
Thank you very much, Validate, for having me.